let me repeat some basic things. I didn't show the photo of Carlo, who's setting up the camera and is putting up the videos. Carlo, is the talk for today, the PDFs, are they online? They're already online. Okay, and the videos from last week? So you see, he is faster, not we. He is faster than the promised week. Again, if you need to send an email, please send it to assistant@Rost.lab. I'm going to be here before the talk and after the talk. So if you want to talk, have any question, please come and see me. CHE, Center for Higher Education Development, is a ranking of universities in Germany. The survey for that starts today. Please take that survey. It's in the interest of the university, provided you like the university. But it's also in your interest, provided you like the university has a good ranking, and provided you get a degree from that university that will help you in your CV. So it's a round circle. That is the website where you can take that survey. The more people take it, and are not negative, the better. Okay, let's just recap. So some of these slides come from Mark Punter, others from Andrea Schapperhans. Genes are the thing that is common to life. There's the flow of information from DNA to RNA to proteins. Proteins are the machinery of life. So in many ways, that's the flow of information. That's also the way it works. It's the central dogma that translates from the information content from the book, from the library, into the machinery that makes the structure, that makes the function. In the course of this lecture, we were mostly concerned about how a stretched out string adopts a unique three-dimensional structure that then has a certain function. And again, you can imagine this like a pearl chain, where you string together different colored pearls or different sized pearls, and you essentially have 20 different colors or 20 different sizes to choose from. Those are the amino acids, and the way chains differ is by the length and by the composition of these 20 units. There are about 20,000 proteins in human. About 55 million proteins are currently known. Length, again, is between 35 and 35,000. The vast range of length is somehow implied in this drawing here. Very, very different scales. And on the most right-hand one, these gigantic structures here, they're even scaled. They're even bigger than that. So one of the important inventions of, if you want, of life is to create or to separate between inside and outside. That's the idea of a cell. Now, in order to build a cell wall, and imagine this like a medieval town or something like that. That's the outside. You have to put a wall around it. You could put proteins as a wall. I say proteins are the machinery of life. Essentially everything that happens in there is done by proteins. So why not use proteins around the wall? Any idea? Any guess? So let me make this point. Very few people in this room will know the answer. It's very unlikely that to these type of questions that I ask, there is somebody who knows the answer. In this particular case, maybe somebody would know the answer. But for most of my questions, they're not of the type anybody in the room knows the answer. Sometimes I know the answer, not always. Science is about getting an hypothesis, developing an idea what it could be. And these ideas could be wrong. The scientific procedure is finding ways to shoot them down if they're wrong. That's what you have to learn as a scientist. But it begins with just coming up with ideas. So become more active is what I'm asking for. So the reason why we cannot use proteins to essentially shape the wall is because proteins are dissolved in water. What surrounds the cell, what is inside this, what is outside, is essentially the similar material as inside of the cell. That's where the protein fold. That means they sort of fold means they adopt a 3D surface or a sphere that is the smallest possible surface, but somehow they can be dissolved in water. Now, as a wall, there's a completely permeable wall. It's not a good idea, right? Just imagine you have a wall around a medieval village that is completely open. so it's not a wall. A wall needs to do some functions. It needs to keep something inside, it needs to keep something outside, and actually we need to regulate the flow of information, or that's the way as a computer scientist you may see it, the information of material. In the medieval village, you do this by having gates, and there's somebody who controls the entry through the gates. Slightly different here. I'll show you some examples. But so we need something in this wall that really protects any water kind of solution. And the way it's done is by lipids. So that is this green wobble thing here. Essentially all lipids. Water cannot get in. At the same time, these lipids somehow allow material to sort of move around. Here are examples for what these lipids or these surfaces of cell look in a little bit more detail. And that slide here comes from Natasha Wood from Cape Town, who used it for a talk that she just gave at a meeting in Dar es Salaam. So that's the surface of a cell, and on the surface of the cell you see proteins sticking out. And these proteins are like flags. That is the kind of thing, you could imagine it in the medieval village analogy. Those are the gates you see from far away. That's where you dock, if you want to enter, right? And in this particular case, the thing that wants to dock is the HIV, the virus, the glycoprotein, so-called glycoprotein. It's a huge aggregation of proteins that contains the information material. And again, the viral membrane, this is the structure that in fact comes in, and it comes in through this gate here. This gate is made open by a watchdog, so to speak, by a protein. And in this particular case, the virus fools that protein and gets entry. One important aspect for this particular story here, for the virus to enter, is that there are sugars put onto the surface that can prevent entry or allow entry. So somehow this sugar coating protects you against entry or allows you to enter. Another aspect of structures that I briefly mentioned already is there are sub-fragments. So you can look at this huge protein structure, and you always, or very often, will see substructures. Two types shown here, the helix in red, and the strand/sheet. So one is a strand, two together is a sheet. In this particular case, you have three strands together forming a sheet. These are recognizable substructures of the three-dimensional structures. In fact, they are stabilized by hydrogen bonds here. For the strands or the sheets, it's between this yellow and that yellow. For the helix, it's within that helix, and we get into more detail of that. Now there are other components of structures. I said already there can be 35 residues to 30,000 residues. And you can immediately imagine that if you have such a coarse or wide spread, you cannot somehow do it without some sort of idea of repetition, some sort of idea of unit that is beyond the amino acid. I showed you helices and strands. They are substructure. But we need a little bit more than that, it turns out. And that little bit more than that are domains, so-called domains. Now let's get a little bit back to one detail about the way proteins are built from the genetic information. The genome, the human genome, I said, has 20,000 proteins, and in fact it has 3 billion bases, nucleotides. Most of the human genome is not translated into proteins. I said again and again that proteins are essentially machinery, they do the life, yet most of our genetic material is not there for coding proteins. Why? In fact, most is not the right word. 99%. Has reasons that are beyond us today. 99% of your genome, we still don't know what it does. We begin to understand some of this 1%, and some of this 1% is in fact the proteins, and that is what we talk about. Now, here is, let's call it structure of a gene, the genomic material. There is something here in green where the reading starts, where the translation into a protein starts. Then there's an exon and an intron. And that is simply the lingo for exon. The red parts here, they eventually will become a protein, and the introns will be taken out, so-called spliced out. They will be ignored. whatever the reason of those intrances. And this particular example, and that is here the end. Stop reading, it means. So what will happen first is that RNA splicing will simply take out the blue ones here, the introns, will only keep the red ones. That creates the gene product, the proteins. Now, originally that was thought about one reason why there could be domains. Just imagine that each exon here codes for one protein. And for the very first proteins that were discovered, or the genes that were discovered, that was true. Turns out it's not true at all. But what we do observe is, say we have a bacterial protein here, A in green, another bacterial protein, B in purple, and another protein here, C, in this particular case is also in bacteria, that has both. This is the kind of scenario when you begin to suspect, that could be that C is actually built up of two domains, two regions that can work alone because they exist in this maybe different organism even. In this example, in fact, that is a different organism. These two proteins exist separately. So another thing that you can actually suspect now is that these two proteins here in that organism will work together. They will bind. And that in fact by P2, the bacterial organism that has them as one protein has an advantage or created a faster way to making them bind simply because they are already together. They don't need to find each other. And this in fact is often the case. Again, such a thing reveals the idea of a domain. Let's get back into the visual idea of domains. So here you have an example where you see that something here is similar. You have a two-domain protein, and one of these two domains here somehow looks similar. In this example, you see a little bit of the same. You see this beta strand in yellow, the purple helices, and that is part of what is in here more than once. And this image, that by the way is from Christina Rengo from London, I didn't put her name on it, I'm sorry. Here you see an aggregation of many of these units. The way Christine, who builds domain databases, and people such as she think about this is there is a core, and around this core you have embellishments. You have just additions that do something on top. It's like a core unit, and you add something more to this core unit to do something else with the one on that side. But the basic function you can do with that one. That's the idea. And already from the quick looking at it here, so each of those would then be a domain, a unit. Yellow triangles here, the purple dots, and you see that many proteins here have many of these units. It's not as in the examples that I showed you where you have two. Many more. How can we guess the domains from the sequence? Well, from what I just told you, let's just assume this following case here. We have these 6 proteins, A, B, C, D, E, F, and what this line signifies is simply the start of the protein, the end of the protein. What the overlap between A and B here signifies is that in the region in which they overlap, the black lines somehow is similar. Okay? If I plot these images of those, you would recognize that they look similar. And we get, toward the end of today's lecture, we get into how we can actually do that. But now, let's look at this example. So we have this little bit longer protein. We have 2 proteins, B and C, that essentially are very similar. They align. Align means I superpose them in a way in which I recognize that they look similar. And then I have D, E, and F, in particular D and E. They in fact do not align. So there's absolutely no similarity. So the black A and the black D, or the black A and the black E clearly do not have an overlap. And then I get into F, where F in fact has an overlap with this, and then F has an overlap with these two. When you see something like that, you may suspect that there are some lines that I can cut, and that we in fact have a story like this one here, where we have a domain overlap. So where this could be one domain, this could be another domain. These regions in between here, it's unclear whether they're regions in between domains or very short domains. Now, this again, the same holds here. I don't have anything that is similar to that. That may be a domain, and if I just had enough proteins, I would find something that is similar to it and recognize that it's a domain. Or maybe it's just a hangover. Same here. Well, here I have two, so most likely that is actually a domain. Now, in fact, very often this kind of situation is found, but sometimes the situation of the overlap is more like this. And in a situation like this, you immediately recognize, while I can easily put a cut here, saying there's two domains in this problem, here I can no longer do this. And more this kind of situations arises, and I'll get back into more detail on that one later today. Prokaryotes are organisms that are simple bacterial organisms that don't have a core. Karyot is the Greek word for core. In fact, the nucleus is what it is called today, and the nucleus contains all the genetic material in a eukaryotic cell. Okay? Bacteria, that's where we sit, but that's also where yeast sits. Yeast is a single, so baker's yeast or brewer's yeast, what you get in the Bavarian beer, is in fact a single-celled eukaryote. And all the other animals are eukaryotes, those are bacteria. They don't have a core, those do have a core. The core contains the DNA. Now, I'm going to run by you a few statements or questions. And I'd like at least for some of those, I understand that most of you have very little knowledge about proteins. Just guess. So the first question, longer proteins are rarer than short ones. I heard a yes. Let me observe that this is not trivial. So it's not just probability or anything like that. So we could imagine a Gaussian distribution. And in any Gaussian distribution, as you all know, both extreme ends are alike. It's completely symmetric. So in that particular case, longer ones, if the distribution were Gaussian, longer ones would not be rarer or more common either way than shorter ones. In this particular case, somehow you saw the distribution from 30 to 30,000 sort of gives it a little bit away. Yes, longer ones are much rarer. It must be because somehow the units are shorter. Otherwise, short proteins could not be 30 residues long. Another idea that has been around for a long time is that proteins in prokaryotes are shorter than proteins in eukaryotes. And that essentially goes back to a common misperception about simplicity. We think about us as, or maybe some of us think about us as on top of some, we are the kings of evolution, is what some people see us. Now, kings of evolution means we are the most fascinating thing that evolution has ever produced. The most intelligent, the most advanced, the most complex, and somehow longer is more complex. So that is, I believe, more than anything else, this analogy why people started it. It is true that prokaryotes are a little bit, prokaryotic proteins are a little bit shorter than eukaryotes, but it's more not true than true. I'm sorry. Here's an example, and that is bringing us back to the issue of the Gaussian. What I show here is the length of a protein, the number of, it's called residues here. When amino acids are put together, they form the peptide bond, they're called residues. It's the same thing. Percentage of proteins, so simple distribution. In blue, you have the eukaryotes, so yeast and we, and green and red both are bacteria. Both are prokaryotes, so archaebacteria and regular bacteria. Now the first thing you see is, as I said, there clearly is no symmetric Gaussian here. There is some average somewhere, but actually the average is so broad that talking about mean and median here does not really capture the story. You see that if you came with some sort of region that you would call the average, that in fact blue, green and red are very, very similar. You also see that green and red are higher. So that's the overall percentage of the distribution in this region, meaning that blue in fact has a longer tail. And this longer tail makes the statement that I had before true. So they are a little bit longer. Oh God, I'm sorry for this animation. We skip it. Okay, let's get back to what we did so far. How can I guess domains from just looking at sequences? From looking at structures, and I'm not going to go back. From looking at structures, this is essentially what I did. You stare at this drawing, the cartoon that I showed you, and you somehow see units that look different, where you have a higher density of contacts, where you have a shape that looks more like a sphere, where you have the feeling that in fact you connect two fists, for instance, in this particular case here, with my pointer in between, then that would look like two domains. On sequence, the story becomes slightly different. And that, in fact, you use exactly the kind of technology that I showed you before. But very often biology simply begins by using whatever information it has. So one information I said, when we know the three-dimensional structure, when we can look at the object, we can somehow see the domains. So we come with a protein for which we do not know the domains here in black, from begin to end. And we find in blue here a similarity to a region for which I know the three-dimensional structure, for which I can look at it and say those are domains. Okay? So if I can do that, then I can simply say, okay, since I do not know for the black one here the structure, but it looks similar to the blue one, so I'm going to cut here. This is too short, I will ignore it from now on. And in the next step, and again, I'm sorry for that, ooh. In the next step, there's a database called PFAM. Protein family is what it stands for. PFAM is done by experts who look at sequences, stare at them, and do whatever the experts do in order to deduce that this looks like a domain. I'll show you, essentially it's getting back to what I showed you before. I'll show you the example again, that you find a region that overlines in one protein, the protein C, where you had a long region, and in another organism you had A and B, and they aligned to C, but were separate. So you had two different proteins. In the other organism you had one protein for both. That's an example how you can find domains. And that is one way in which PFAM, for instance, identifies these domains, annotates them, puts them in a database. And at this point, they have about 10,000 of these domains annotated over the last 20 years. The way this is done is one at a time. Sometimes the annotation of a single one of these domains takes a day. Sometimes it takes a week. When you see a similarity to some region like that, you cut again. Now we get into the third level. In the third level, and I don't know, some of my slides are really messed up. I'm sorry for that. I give up. At least this one. Yeah. Oops. Never do animations. If you didn't know yet, please avoid animations. So in the third level is exactly the kind of thing that I mean. You cut here, throw that one away, that left you with this fraction. You cut here, so now you have this one and this one to match. Say in this one you can't find anything, and in this one you have exactly the situation I showed before. You have a protein C, a protein A, and B. Possibly in one organism, these two here, and another organism, so you can deduce that these in fact will also be two domains. So you cut that into two. And in this particular example, you would then have 1, 2, 3, 4, 5 regions, where for some of these, for this, and these two, you have some explicit information, some explicit suggestion that they form domains. For this one, that may be cut in the future if you have enough new sequences, or that may in fact be a domain you don't know at this point. And this is essentially the way it works. What we find out when we do this on a large set of proteins, I show a number of domains here. Green is proteins for which I know the structure. Blue is this procedure applied to millions of proteins. What you see is that green and blue differ very much. And that is because green are proteins for which we know the structure, I said. And it's much easier to determine a structure of a protein if it has only one domain. That's why this is highly biased here. In the blue distribution, you see that 1, 2, 3 domain essentially is a similar fraction. And remember, we made mistakes, so sometimes we cannot continue to cut simply because we don't have enough information. So this clearly is an overestimate. If anything, then the distribution will move a little bit in that direction. But most proteins, then you see here, clearly have more than one domain. And most domains are about 100 residues long. Okay, let's get back into bear with me. Let's compare proteins now. So, before we compare proteins, I want to begin with another notation or wording. So, I showed you both these images, that one too. In fact, those are two different subunits of a same protein. Those rods are helices. These are beta strands. Another protein here, a cartoon. So there's an annotation about 3D, 2D, and 1D. I'm sorry for the slides. I really do not know what happened. Because there's something missing here. 1D is missing. Oh, here it is. So 3D is simply for every single atom, or every single amino acid, every single pearl on this chain, you have its three-dimensional coordinates. 2D, you take out this protein, you take two protein, you stretch it out again, and for every residue, for every point, you ask, how close is that to all the others? So you build the entire distance map. That's a 2D representation. For every residue in your protein, you look how close or far away is that from all the others. 3D, 2D. That's clearly a reduction in complexity, right? One dimension less. Does it contain the same information? No. What is missing? It's true. It does not contain the same information. But what is actually missing? The helix. Well, helices are stabilized by hydrogen bonds. Hydrogen bonds are bonds. Any bond is, of course, in a distance map. Any binding, anything that glues things together is in the distance map, right?
Information about the structure of it.
So you say the information about the structure is missing, but which information exactly I'm asking?
The 3D shape, how it folds.
So let's just get to the answer. Yes, I lose some information. But let's get to what exactly I lose. So you say what I'm losing is the way it folds. Somebody else already had this point about helices here. Helices are stabilized by hydrogen bonds, and that information is in there. Now the way it folds, so the way the trace goes here is in there, because at any turn, any angle is written here in the distance map. In principle, if you have a 2D map of a 3D object, you can turn it back into the coordinates. Maybe not trivial, but doable, because for every single point you know the distance from this point to everything else. And ultimately, there's only one solution. That's not true, and that's the part of the information you lose. There are two solutions, and that's the mirror image. You cannot distinguish between the right solution, the 3D structure that you have, and its mirror image. But that's the only thing you lose. That's for computer graphics. 2D is a complete representation of the 3D information, except for the mirror story. Okay? Again, of course, that depends on the level of resolution here. So I talked about a distance map. If you just had a sort of gray, black and white map, so not a distance but a contact, so something is close, yes, no, then you would lose information. But as long as you have really the actual distance to every other point in your object, you can completely define the three-dimensional object, except the mirror image issue. Now let's get to another extreme reduction here. Well, it's the same. I go from 3D to 1D to 2D, and from 2D to 1D. I reduce another dimension. So rather than having the pairwise information, I now simply have a string. And for that string, I, for instance, know the sequence. Or I know at every single position here, at every residue position, the T is in a particular secondary structure, E means extended, means beta strand. And that happens to really be the strands that you see here. Now, if I just looked at the E's here, so I know for every single pearl the secondary structure it is in. That's the 1D information. Do I still have the 3D information in there? Or did I now lose something essential? It's great that you dared before. Dare again.
We don't lose any information.
From 2D to 1D? So from 2D to 1D, now getting back to what you said before. You said what I'm losing is the fold, the way it folds. And in 1D I don't know how it folds. I know, okay, or somebody else said helix. Well, knowing that this is a helix here does not mean that I know how this helix is positioned to that strand. Does not imply that I know, well, I know that the helix has a certain shape. Okay, I can build up one helix if I know the secondary structure. But once that helix ends, I don't know whether I go there or there. That I don't know. Yes, I find the strands and I know that the strands bind, but I don't know which ones. The bonds that bind, it could be, I'm not quite sure that I see the way this one goes here. It goes up here, out. Okay, we go here, back in, here, here. Okay, so here we have two strands that are next to each other. But it may be, in fact, that this one binds here, or this one binds here, but it may also be that this one binds here. And that, in fact, exactly defines how they fold. So just looking at the 1D structure, just knowing the E's, just knowing the H's, does not give me that shape. So although this is one dimension less, and that's also one dimension reduction, this one crucially removes important information about building up my three-dimensional object. I may still have important information, and in fact, in the last column here is another aspect of that information. That other aspect is so-called solvent accessibility, and that essentially means, imagine a protein is sort of spherical. Then there are some residues in the inside, and there's some residues on the outside. And you immediately see that will help you to get back to some of that information of the shape. So now you know that's a helix, that is a strand, and you know that this strand is on the outside. You still have a lot of possibilities. Maybe there's something else that you need to know. But that would already help you. So you already see that with 1D information you can possibly build it up and you can get a good guess. But still, this step here has an essential loss of information. And again, another point, secondary structure is 1D. It's essentially writable as a string, although it is stabilized by contacts. Okay, how can we compare three-dimensional structures? So we look at these things from looking at the residue version, at the atomic resolution. It's not that easy to see that blue and red is similar. Looking at it here, you even unused to structures, now you have seen it so often, you even begin to suspect that red and blue is similar. So our eye is actually very good at doing this. How can we compare three-dimensional objects? So again, the task is to simply see that red and blue is similar. How would you do that? Has nothing to do with proteins, in principle. We have three-dimensional objects, and I want to compare them. Any idea? Okay. Well, somehow you have to come up with this idea, and for children this is remarkably difficult, to see the main aspect of a shape. When you match shape, essentially one way of doing it is you put it here, you put it there, and you ask which of these two is most similar. Now, how can you answer most similar? Well, the simplest one is simply you define some points here, and you see whether these points, the corresponding points, match. But it's immediately clear how they match it. So in this particular case, you would have 4 points. 1, 2, 3, 4, and you would see that these 4 points match and cover more of the surface there. There's a difference. Now, that's the first issue. What do you do? So in the first case that I had here, on the right-hand side were the objects that I tried to match to. In my first example, although the shapes are very different, the reference points clearly match. So what do I do if these reference points no longer match? So one of my reference points lies here, right? And this one no longer matches at all. That is one problem that we have. And then one way of doing it, you see whether you have different ways of orienting it. So now it's no longer a solid object moving onto a solid object. Now you rotate. And you see what's with my reference points. Okay, my reference points doesn't match, but what about the other 3, or whatever the other number of points is that I have? Is there a better fit? In this particular case, so I'm going here to 8 reference points, and then I can compile, once I have those 8, I can compile the difference. So I do my 8 points, and for every single point I simply ask, between these two shapes, bless you, between these two shapes, what's the difference between the point, so that's the match state, between here 4 in green and 4 in red, and we do this by a simple radius difference, normal distance, a square root difference, right? Anybody in the room who immediately sees what this formula, if I measured my difference between 2 objects by this root mean square deviation. So I compile a square of the differences and I sum up over all the reference points. Anybody immediately sees what is the implication of that formula? The variance. Yes, yes. It's totally true. The variance has the same idea behind it. But I meant more mathematically, or more in terms of looking at 2 objects. Let's go a step back. Is there another way in which I could compile it? So you're totally right, this is the variance. Why is the variance defined that way? Is there any other way to define it, a distance between two points? Okay, it's the vector distance. We grew up in math with that. But could you imagine another way of compiling distance? For instance, exactly, you could measure the angle. That's one idea of a distance. So you could argue, okay, I have a projection and I look at the sine of the angle, or whatever you want, but that would be a different way. But I meant something much simpler even. I mean, look at this formula. What's the sum you need? Yeah, you need the sum of all the differences. That is not the part that I'm talking about. But in this part here, how can you simplify that part of the formula? The angle is clearly a solution, but even simpler. It's like that, exactly. But how can I simplify that formula, or how can I measure distance between 2 points? That is the standard deviation, that is the vector distance. We have all seen this formula and again, and it comes to us intuitive. But mathematically, there's an operation here that makes it's not the simplest solution. So let's just get rid of the 2 here, the square. Right, yeah?
Absolute difference?
Exactly. So just getting rid of the 2 alone is not enough, because then we suddenly have an orientation, and we don't want that. We want an absolute value, and we simply do the absolute value. When the squaring, one effect of the squaring, and that is actually the same for variance, and in the statistics it is a wanted feature. Now, but one thing that you have to remember when you see a square like that is that it implies it will zoom into big differences. So the sum here will be absolutely dominated by the largest value, simply through the square. If we summed up the absolute, that would not be the case. We could do that differently, and in fact there are solutions that do it differently, because in some cases this is not quite a wanted effect for proteins. But okay, back to the story here. This is, we all admit, a relatively simple way of doing it. Root mean square deviation, and applying that root mean square deviation, you simply can compile the difference between 2 objects. Now, what you have to do is you first have to find the corresponding point, then you do the superimposition, and then you compile that root mean square deviation. Now there's something else. These 2, for your eyes, immediately you fit them, right? At the same time, you immediately see the difference between the 2. And that, in fact, is not that trivial. If you just did these corresponding points, you would see a big difference between red and green. Rightfully so, if they were proteins, because they are different objects, different sized objects. But you immediately see that we need another operation here. We need some possibility of a blow-up. Back to the proteins. That's exactly what we do. So now we have another issue here, and that issue is the difference between a global match and a local match. What I mean here is, say I gave you these 2 objects. You immediately would argue, or you immediately see the similarity of a person or of a robot or whatever you call that thing. So I'm a great artist, and that is as close as I get ever in my life to drawing a human. You immediately see that they are sort of analog representations of each other. Now, if you went back to what I just did, you find representative points, you do the optimal superposition, and you count the root mean square difference. You see this would be horrible. In fact, you would never, if you did that operation on the entire object, you would never see the difference, the similarity between these 2. They would look totally different, because the relative parts here are very differently ordered. The way you do it in your head is you take it apart. When you align the red and green, you actually align the pieces. You see that there is some order in the event, you see there are similar parts, and that makes you see the similarity. The local similarity would begin by by seeing a unit, such as the head here, being similar. A global would do something like that and would look very different. But now we, in fact, can immediately imagine how we can establish a similarity here. Just like with this robot, proteins are made of parts, so I have to cut it. And then I simply align all the parts. Now, one issue with this cutting business is, when do I stop? One extreme way to stop would be at the level of the amino acid. But that would be nonsense. An amino acid does not have information about three-dimensional structure. So then you could argue, you cut at the level of secondary structure. Those are, I told you anyway, substructures, right? But secondary structure, again, now we're back to the problem. When I go from 2D to 1D, I lose information. So if I cut my object in pieces and look at the 2D aspect of the object, then I no longer have the 3D information there. So that cannot be the solution. So I need something about the 2D map, something about the fold, something about the three-dimensional structure that is more than just secondary structure. So I have to be careful with how I cut it up. How can we decide what is a valid unit, how far I can cut? And in some sense that depends on what I want to do. What I want to do here is I want to compare two objects. When is it too small? And in some sense you could argue, well, too small, if I want to compare structures, too small will be when you get to a unit that matches everywhere. Say you have 1,000 proteins, 1,000 different structures, 1,000 different shapes that you believe cannot be matched to each other. Now you come with a new shape. And if the units you cut it back into map to every one of these 1,000, then you cut too much, right? That's very clear. Because then you have no longer a power in the comparison. You have no longer, by the definition that you have 1,000 that are not similar to each other, going too small, matching all of them would be too small for the comparison. So we need something that matches only in some of those, or maybe only in one of them. Now that brings us to the simple statistics. You, in fact, throughout biology you will have something like that, an expectation value. You will see something in the background, here in blue. For instance, you take secondary structure. You will match some part of a helix in almost every single protein. In some better, depending on the length, and some a little bit worse. Those are beta strand proteins. That's the background. That typically is the signal. That is the similarity for which you have established something. So in this particular case, would, for instance, be a domain. If you see something such as this, then you're on the right path for having cut not too far and having cut in a somehow reasonable way. How can you now compile a value that reflects the trust you put into your match? Well, it has to do with two aspects here of what I show you. One is how red differs from the mean here. So how far away from the random background you are. And it has to do with how broad the variance is here, how broad the random background is. In this particular case here, red is a clear signal, and everybody sees that. If I had plot the blue such that, in fact, the variance would go all the way up here, and red would be totally encircled by the blue distribution, then although red may be away from the average, it still would not be a very strong signal. And these two aspects, in fact, are part of what defines a good match. So again, now we do a 3D comparison. The way we do that is we look at our object, we cut, we compare, we compile an RMSD, and we move these objects around in the space. And essentially that's all we need. Is there anything else other than Cartesian? So Cartesian, simply the coordinates I put into, and in some sense that your colleague already sort of hinted in that direction. Instead of Cartesian, we could also look at the angles. we could look at essentially, you could place yourself on on a particular point here and define the universe with respect to where the next point, if you were number 6, how you get to 7, how you get to 5. And you can define the shape here through local vectors, right? Through the local angles. And those, in fact, is another way. And I will show you that way too. And here's yet another way. You can, in fact, define this object as a matrix. I already told you that 3D to 2D, if you have the entire distance map, is containing completely the same information. And mind you, I'm not talking about proteins here. This is just objects. It's nothing specific, except for the part where we get into the unit issue. But the unit issue, the examples that I showed you is a robot. So matching, seeing, recognizing environments in a computer graphic program is the same task. We also will have to see what the units are. The only difference typically is when you write a game or when you recognize an environment, then typically you have a better idea what the unit is, how far you can cut. But then if you look at pictures of the Earth and you do not know at which distance of the Earth they have been done, you will also see that even the scaling issue, even there the cutting the units is not a trivial issue. But somehow it's the same thing. And again, so 2D map contains the same information. That means I can also represent here my 3D information as a 2D map. So 1 is next to 8. In this particular case it becomes a contact map here, but I could put more gray levels in there. So define exactly how far 1 is from 8 and do this for the entire matrix. Once I do that, I can now compare two objects by comparing these matrices. How can you compare two different matrices? Well, obviously there are different ways of doing that. One way of doing that is you just map the entire matrix and look at the difference. That would be like taking these two robots, comparing them just like that. I said you have to cut. Cutting in this universe here implies that you take fragments out of that matrix, right? So it's just the square, the body of the robot, for instance, which would be defined as some square in here. Okay? So far, so clear? In principle, yeah?
How many matching points are commonly used?
So again, that depends on your problem. So the rendering of your 3D graphic in your computer would be defining the answer to that. In a protein, typically you would argue, so what is the unit of not comparison, but the unit at which we plot. I showed you examples of proteins where you had an atomic resolution, and then I said in this atomic resolution you cannot really see similarities. So atomic may be too much, but in some cases atomic is what people do. What I typically showed you when you began to see things is when we had the rods, or in fact even in cases where you had essentially 1 point per pearl. 1 point per pearl in the example of proteins is 30 to 30,000 is the length. He said that no, I didn't show you that, I glossed over that graph, but most domains are about 100 residues long. So typically the answer to your question then would be that each of these would be 100. Yeah? That's the ballpark issue. In some cases one of them would be 3 domains long, one would be 1 domain long, so this one would be 100 times 100, this would be 300 times 300, and then you essentially look at, I don't know, fragments of 50, 60 cut out here, and you move the 50, 60 around and try to find the 50, 60 of the left in the right. Okay? So now let's get back, so far, nothing I said really has anything to do with proteins. Let's get back into the biology. And the work that I present here, the slides come from Patrice Kuhl at UC Davis. So in proteins, it's the same thing. We now, in this particular case here, have 2 fragments, this and that. We want to map this to that and that to that. By now, you have seen enough proteins to immediately do that. It's a simple 3D matching problem. So you identify equivalent positions, you find the superposition, and then you compile the RMSD. Now, in this RMSD, in this particular case, RMSD, this stands for C alpha, which essentially means 1 point per amino acid. That's the answer that I gave you. 2.8 angstrom. Angstrom is 10 by the minus 10, so this is 0.28 nanometers. And that essentially, angstrom is a value that is taken in the world of proteins. Simply, it's 0.2, 0.3 nanometers, but it's typically angstrom is what is used, and I will get to the angstrom. Gives you an idea, 10 by the minus 10. So you have these two examples here, and both of those, the root mean square deviation. So the sum over all the reference points are the amino acids, the sum over all of those is 2.8. 2.8 is in fact very similar, so the blue matches in a similar way as this helix. You also immediately see that the helix is in fact not quite that helix. It's not that easy to see that here. But that gives you some idea how much here does not match. Now, here's an important thing to see. If that were one protein, then A is similar, protein A and B. They are similar to each other. B is similar to C. But C is not similar to A. The triangular inequality does not hold. So this is not what that means in terms of math. This is not a metric. The comparison of these three objects here is not under a metric. And that is in fact very important. It also does not mean that A and B are not A and C are not similar. I just can't tell. Okay? So now we get into particular algorithms in which we can do that. And essentially what these algorithms do is find a relatively fast path to solving whatever kind of problem I had before. Now, ultimately, why do we need an algorithm for that? Well, because when we get to hundreds of points, it gets complicated. So there is no longer a trivial solution. In principle, the idea is clear. How to put that in a computer may not be that simple, or there are alternatives, and ultimately the alternatives have to do with it takes time to run this comparison. Here comes from Taylor and Orengo. From Taylor and Orengo, essentially what they do is they describe every residue I in protein A and residue K in protein B through a vector point, look at the differences as before, and put that into a dynamic programming environment, a double dynamic programming environment. The double dynamic programming environment essentially goes down the path of residue by residue and asks what does that local vector environment fit best in the other protein. Puts a point how well it fits. And then it backtraces and sums over all, finds the optimal path, the highest path of match through this. And that essentially is the same formula as before, adapted to the situation where now this is distances and pairwise matches. Algorithmically, this is way more complicated than before, but in principle, it's the same problem that I showed you before. The problem is that doing this, just like in the conversion from 3D to 2D, you lose the directionality. Here, DALI is a 3D alignment from Holman-Zander that goes slightly different. So that one, in fact, goes directly into the matrices, the distance matrices that I showed you before, compares them one by one by Monte Carlo algorithm. So simply, again, now what you do is you grab out subfragments. You ask whether that subfragment by the Gaussian, the red versus the blue, so how much is a significant match deviating from the environment, and you compile all possible matches. Okay? Here's another one that in fact came from Munich, from Bürzel and Zimmer, LMU, he's now in a company, Fabian. That idea is I built I take my protein structure and I build Voronoi trees from it, or Voronoi surfaces from it, Voronoi objects. So those are local shapes, and I simply compare these local shapes. Still, like the idea of reference points that I showed you with the sphere and the square, but now we no longer have two-dimensional sphere and square. Instead, we have simply three-dimensional objects, little cutouts, Voronoi surfaces or two-dimensional objects here, but they are no longer simple squares. And now I take all of these and do the same on the level now, not here, the entry to that matrix here no longer is the amino acid residue. So back to your point. So here you simplify the number of objects that you compare. So Voronoi surfaces rather than amino acid residues. So protein has 100 residues typically, or structure has 100 residues. Here we would have 50 of these Voronoi surfaces and compare those. Reduction of half in the complexity, but very meaningful because the objects you actually compare are more revealing than the residue level. That's another way in which we can do it. Other than that, you simply do the same matrix comparison. Okay, you simplified the problem by doing the right geometry. You solve the issue essentially by geometry. The algorithms for the comparison is the same as before, but at lower level of complexity here. What else could we do? Well, one issue here, and that deep is biology, is that binding substrates, in this particular example here, we have actually the same protein. You may argue that to you, on that side where it's red here, it looks very different from the side where it's green. And it does. The way that happens is it binds. Essentially, binding means you clamp, you close, you grab. Just the world of protein structures is very intuitive in some sense. A lot of the changes are such that you can actually see them. So grabbing some substrate, that's what it does. Now, the trouble of course now is, if you had this to start with, the similarity between green and red is not that trivial. It's not clear whether that in fact, this sort of curved object is in fact the same as this one here, or a different protein. You need to know more. So in this particular case, if you knew that this is in fact the bound state and this the open state, then everything becomes simple. But if you cut out here the binding part of the substrate, and you just had the red and asked whether it's similar, then you may conclude it's not. Those are in fact issues, and that brings up another issue. There is the global alignment in this particular case. Global means you in fact force from begin to end red and green to be aligned, to be superposed. And there's a local alignment. The local alignment in this particular case here is essentially one of the binding sites. And that brings us back to the robot idea, whether I force the entire red robot and the entire green robot to be aligned, or only the heads and body shapes. That's the head and body shapes only, and then you immediately recognize an important similarity between the two. There are many other methods that do structural alignment, or that do the comparison of proteins, and ultimately most of those differences have to do with how the algorithm is done in detail. Now that ultimately has to do with CPU. So what are we talking here? The situation is that typical programs as they exist today take on the ballpark of an hour for comparison. So we take two proteins, we find out that they match, takes about an hour. That may not strike you as a lot, but the fact is we have 100,000 proteins that we have to compare to, or 10,000. And you immediately begin to see that there is a CPU issue, and there may be an advantage in fact doing some of these steps faster. And that in fact is the background for many of these other programs. Now sometimes when speed you typically gain by simplifying. Simplifying typically means you lose accuracy or precision. You throw something away. And there's a trade-off between these two values. Rachel Kolotny, Patrice Köhl, and Michael Levitt, who went on to get a Nobel Prize, did a large-scale comparison and found out that essentially many methods have an advantage, many methods have disadvantages, and it's actually complicated to really say this is the best method out there. And that brings us back to another issue, the issue of standard of truth. So I want to see how well my structure comparison method does. Let's go one step back. I haven't said that, and that's a mistake. When I have these three-dimensional objects, again, I take the stretched out Pearl chain, put it into a solvent, it adopts a unique three-dimensional structure. That unique three-dimensional structure is so important because it comes along with a unique function. And you immediately see why I need to compare objects. The 3D structure reveals something about function. That means that if I have a new protein for which I know the shape, and I can match it to that one, that tells me something about what this may do. By matching structure, I can match function. By matching function, I learn something that I may not have known before. That's the incentive, one of the incentives for matching structures. Another incentive for matching structures is essentially if you want to understand how proteins have evolved. I gave you this example where we had one organism, one protein A and B, two different proteins. Another organism that had these as one protein. So if you see something like that, you may suspect that these two proteins interact, I said, but you also may suspect that these two organisms are evolutionarily related. And that in fact, the way this evolution worked is at some point the gene got merged. So you may remember that I talked about introns and exons. So somehow all you need to do, there was a start in that slide of introns and exons. There was a start of reading to translate the gene to a protein. Just remove one of these starts or ends and immediately you have these two connected. That's one way. Evolution is another reason why you may want to have this comparison. And in fact, another important way is to see more details about the function, and that ultimately relates to drug design or relates to defining drugs for certain diseases, for certain proteins that go wrong. So all of that requires methods that can do the comparison. We have many methods out there, so that creates a high incentive for comparing them. But the comparison itself is not trivial. How could you, in fact, come up with a standard of truth? Well, one way would be that you have a bunch of biologists who build a data set, and that is the standard of truth. So they really know these two proteins belong. They are similar. They have a similar function. They have a similar structure. So my method should find those. That's one way of doing it, and in fact that is historically the way that has mostly been done, and that in fact is largely behind this ROC curve here from Rachel Kolotny's paper. In this ROC curve you somehow see a bunch of methods, and you see that there are differences, but you also see that there is a high density of many methods that are all pretty much up here. That's the only take-home message. Different methods, there are many good methods out there. Let's get back into the number game. 3D comparisons. How many are there? How do we see that? How do we see the universe? One way of seeing the protein universe is it's this globe here. And each protein structure is an island. Okay? Here some of these islands are green and some of them are white. The idea behind this coloring was that white islands are those for which I do not know this three-dimensional structure, and green islands are those for which I in fact experimentally do know the three-dimensional structure. And the unit of that universe then would be the domain. So this sort of cutout symbol here, that is part of this one. So, and that unit we can see from sequence, and essentially every one of these islands is a domain. Now, André Lupas from Max Planck Institute in Tübingen is one of the people out there who has this idea that life of proteins evolved as slaves to RNA. The original form of life was RNA molecules. RNA molecules could not reproduce themselves. They needed proteins as slaves. So they found amino acids or peptides. Peptides are short stretches of amino acids put together in a peptide bond. So two amino acids together, one peptide bond is a very short peptide, the shortest peptide. And essentially they found these short peptide to help them. And if that were true, then we would immediately understand that the peptides we see today, that the proteins we see are biased towards interaction with RNA. We may also see that some of these ancient things that RNA picked still exist. And that essentially is his life's project that he has been working on with many people in the Max Planck Institute for the last 20 odd years with remarkable results. And essentially, again, the idea is take today's structures, stare at them, and find these. I talked about domain, but now we talk about units that are shorter than a domain. That are things that don't really fold real structures, real protein structures yet. This is historically before. We're talking about short stretches that are not supposed to fold spheres, as I said. These are short stretches that are supposed to help the RNA. They could just be this stretched out double helix, or two parts of a helix here, or this type of helix that is an interwound helix, which in solvent, put in solvent, would not be happy, but as a slave to RNA would just be the right thing to do. And we do see them. And sometimes, in fact, we would have shapes that immediately string together, like this one here, immediately string together to remarkable protein shapes here. This one, in fact, is a porin, it's called so-called porin, that makes a pore in the membrane. So this is another way of, in fact, guarding the village. That really is, in fact, a gate. Sits in a membrane, immediately allows access, sometimes gated. This is another shape, the so-called propeller fold, well, simply because it looks like propeller. It's built up of these strands put up together like sheets. The binding site typically is in the center here. So they look at these shapes, they try to see experimental evidence for that these shapes would in fact do what they would have to do in order to serve the RNA, that they exist alone and that we could synthesize them in the soup that existed billions of years ago. And again, they have come remarkably far. Another person, Nick Grisham, at the University of Dallas, simply came to a similar idea of the statement that domains are not the unit by staring at comparisons and arguing that sometimes the similarity between objects implies that this, what is similar between them here in red, for instance, is in fact clearly not a domain. There is something that you see as a repeated unit, and it's not a helix. And it's not a domain. It's something in between. And how long that something, in this particular case here, is this L-shaped two helices, or you can expand it here, the L-shaped two helices together with these yellow strands. They have a particular spacing. And the remarkable thing here, the idea of a domain is a unit that exists on its own. Here, for the two yellow strands and the two helix parts, in gray and red, they don't exist as a unit. But it seems to be a repeated, observed subunit. So that implies, for Nick Grisham, that in fact there is an evolution of these units. You put them around something else. In this particular case here, you just put two of these together, and you have a three-dimensional shape that could exist. That's how we could have evolved proteins. That's what Nick says. Now this implies that rather than this image here, where islands are defined as you cannot go from one island unless you have a ship, to something where you have a smeared out version of the same thing. So where in fact you can sort of go from island to island with the level of water is very shallow. You essentially can walk there. Walk there, in the analogy of protein universe, is by having a fraction of a domain, a short region, you can go from here to here. In terms of the way we think, we perceive, we imagine the protein universe to have been created, these two alternatives are important differences. It remains difficult to really see which of the two is true, but clearly today we think about it more in terms of this view, the smeared out view. For the structural comparisons to operate, for functional comparisons, we think about units, we think about domains, subunits, and we compare them in their three-dimensional structures. And that's immensely important. But we also know that's not the evolution, most likely not the evolutionary unit. In fact, things go on that are on a much smaller scale. Still, in order to classify, we need to begin with these three-dimensional units. And again, for instance, to recover this view here of Christine Orengo, the view that says these embellishments are around units and these units carry important aspects of function. The view of Michael Levitt here, where we have all alpha, all beta, and something in between as three simple, that's the simplest classification. Michael Levitt got the Nobel Prize in Chemistry 2 years ago. That's one of the early ideas he had, simply look at proteins on the level of secondary structure and give the simplest classification. Some of them have only rods, some are only red, some are largely yellow, and some are a mix. And this, you have your three classes of proteins. And these three classes of proteins could then be used at the beginning of databases. Three of those are shown here, SCOP, CATH, and COPS, that classify proteins. And all of those essentially proceed the same way. I'm going to, bless you, go through a couple of slides. The details are not important. The background is simply what I said. You begin with a very coarse-grained classification. All alpha, all beta, alpha, beta. That does not tell you about function. It just allows you to start somewhere. SCOP is done by somebody who can look at these proteins. I showed you now some, and you would clearly recognize more similarity than you did before we ever had a lecture. And if I showed you another 10 lectures of structures, you would simply learn to see some similarities. SCOP has been done by somebody who could just, on this computer screen, where you can in fact turn it as a three-dimensional object, look at two proteins and say, I have seen that before. So you could do that at a point when we knew in the ballpark of 50,000 structures. At some point, Alexei Murzin, who is behind it, stopped doing that. Here he is. But at least for a remarkably high number, he simply could just look at it. It would take him just a few seconds, and he would say, I have seen that protein before. Remarkable feature. And he simply did build this SCOP with that feature in his background, an intuition. CATH, the background behind that, or the person in the background of that, is Christine Orengo. I've shown some slides from her. And Christine, together with Willie Taylor, was just one of the people who built the first structure alignment algorithm. And that method simply looks at an all-against-all comparison of all these structures, cuts out units, the domains that I showed you before, and sometimes smaller units, and then does a classification of everything against everything. But does it still driven by intuition, by human experts who look at it. COPS comes from the Zippel group in Salzburg. It's the same idea, but now the computer method is pulled to the extreme. So we have a classification of the protein universe without any human involvement. So this is completely automated. And I believe I leave it there. That's COP. And I will just skip through these slides. The main ideas, I believe, I gave you, and I believe the differences ultimately between these three approaches don't really matter. What matters are the numbers at the end of the day, the players here, the numbers at the end of the day. So all of these methods today we know structures for about 100,000 proteins. And this leads to classifications that make the point of about in the ballpark of 10,000, 1,000 to 10,000 units. So they are unique, 10,000 unique domains. And everything else in these 100,000 is simply repeating these 1,000 or 10,000 units. The difference between 1,000 and 10,000, remember I showed you the universe of proteins. In some sense, let's call it the RMSD threshold. So do two proteins have a similar structure, yes or no? Depends on when I showed you examples where 2.8 angstrom was the difference between the objects. 2.8, do you call that similar? Or do you call 1.6 similar? And that essentially is the difference between 1,000 and 10,000. So that's the range of unique objects that we have in the universe of proteins. And at this point, the assumption is that at least 70% of all the known sequences can be mapped to this variety, 1,000 to 10,000. For the other 30 or 40% of the proteins, we cannot map them to any known structure yet. So we have to experimentally determine the structure. That is another issue. The way we experimentally determine the structure is we take this protein and make a lot of it, high density. So dense that it becomes like a crystal. Once you have a crystal, you can shoot X-ray beams at it. From the diffraction pattern, you can reconstruct the image. That whole process used to be a PhD thesis when I started in this field. Today, this is something that is doable in a very, very good case within a month or a few months. It depends a little bit. It doesn't work for all proteins because it's not always easy to get a crystal. It's always complicated to get a crystal, and sometimes turns out to be almost impossible to get a crystal. The whole process, how much does it cost? Do you believe? So I showed you these images of proteins. You can just put out any of those here. That's a perfect example here. How much does it cost to make such an image? I mean, not to make the image, but to produce the data for such an image. Ballpark. So you have to grow a crystal, you have to get a lot of this protein, grow a crystal, shoot an X-ray beam at it, big X-ray beam. CERN type of X-ray beam. So it's done at CERN. There's no source in the Munich area that could do this. None in Bavaria. Any idea? Ballpark. Who is happy if I said $1,000 or 1,000 euros? These days almost the same. Happy. Who would say 10,000 is an okay number? Some would say that. Who would feel ouch when I said 100,000? Ouch. Anybody in the room who would still be with me at 1 million or would begin to laugh? So there are many proteins are more than 1 million. And the situation in which we believe that many proteins are done at 100,000 has been reached just over the last 10 years. It used to be much more in the million ballpark, and I guess that the average price today is still clearly over $100,000 for every one of these images. On that note, there are 5 here. So this is between 5 million and 500,000 euro or dollar. So it's extremely expensive, extremely complicated to do that, and that immediately tells you something else. If that much money is spent for that, there appears to be a lot of interest, right? People don't spend that sort of money just because we want to study evolution and it's nice images. And the reason really ultimately behind that is the drug industry. It's not a single drug on the market that has not been going through some optimization process for which we do not have a structure. Every drug starts with a structure. Okay, and that is part of the money that went into this. Part of it is directly related. Any kind of drug design ultimately has to do with structures, changes of these structures. Okay, now that also brings us to the incentive for the computational biology. So part of computational biology's challenge is to take a new sequence, a new protein that has been discovered, and map it to the known ones. That means take the sequence and predict its 3D structure and map the predicted structure to what we have seen before. And that's exactly what this lecture will be about, how we do this. Or to, in fact, take this sequence and map it, and find in the database and look up, find something for which we already know the structure. So that could be a Google lookup, but Google lookup is not that simple because typically your protein may not be annotated by its name. There may be a sequence in the database that is similar. And that is what the first part of the lecture will be about. Thank you for your attention. Are there questions?